I'm Sarah Norcross and I'm the Director of Progress Educational Trust, and I've been thinking about how to introduce this event. And I suppose I sort of boiled it back to thinking about equality in a way, because human biology is older than any equality legislation. And it steadfastly refuses to bring itself up to date and to conform with the legislation. So it still discriminates. And as we know, women's reproductive years are still considerably fewer than those of most men. And according to the Office of National Statistics, the age at which both women and men are having children is going up and up. And while for the majority, this won't be problematic, for others, they will struggle to conceive and they will need medical help. And this will be a devastating blow, particularly as not all will be successful. Tonight, we're going to be discussing both the female and the male reproductive lifespan and how science and medicine are helping us to break through the biological boundaries now and how they promise to do so in the future. And as I said, it's really important that we're talking about the men here as well, because they are very often overlooked. But we'll also hear about people who are even more overlooked in these conversations than the men, and they are the children who are born to older parents. What do we know about them and their psychological outcomes? Now, I'm going to put my hand up and say I have a declaration of self-interest here. My mother conceived me naturally in her mid-40s, and I will let you reach your own conclusions about the pros and cons of older parents and how I turned out. So this evening, we're really lucky to be hearing from 4 leading experts: Richard Anderson, Evelyn Telfer, Chris Barratt, and Vasanti Jadva. Richard is Professor of Clinical Reproductive Science and Head of Obstetrics and Gynecology at the University of Edinburgh's Centre for Reproductive Health. And he's a consultant in reproductive medicine at the Royal Infirmary of Edinburgh and at the Edinburgh Clinic. So I'd like to hand over to you now, Richard.
So thank you very much for that introduction. And as always, it's a great pleasure to be speaking with the Progress Educational Trust on this really important topic. And my topic today is to discuss the background to female aging and fertility and potentially what we can do about it. So the background to this, really the fundamental bit of the biology that underpins what Sarah introduced there, is that women form all their eggs before they're even born. And this is a picture down a microscope of a fetal ovary from a fetus at about 17 weeks pregnancy. So just when her mother is starting to feel some kicks perhaps, and she is already forming all the follicles, which are these round structures containing the eggs here, that will last her reproductive life. And they will stay in this non-growing state for potentially decades, and a trickle of them will start to grow every day right through to the menopause. And of course, that then depletes the number of follicles and eggs that remain in the ovary. And this is a classic graph from the science literature describing that. And what you can see is how it's on an exponential scale, this across the age, with the average age of the menopause here at about 50. And log scales are quite difficult to my mind, so I thought I'd just illustrate the numbers that this really means. So if when you're about 20, the average woman has about 100,000 eggs. When she's 30, this has gone down to 30,000, and when she's 40, that's gone down to a mere 6,000 on average. And of course, half the world are below average. So this is a harsh underlying biology that underpins this. But in the face of this, despite this, more and more women are having babies at later ages. And this is the data from the Office of National Statistics going through to a couple of years ago. And you can see in the blue and the yellow from women in their early and late 30s, and in the green in their 40s, how this proportion of women having babies at these ages is increasing. And indeed, for women in their late 30s, it's tripled since 1980. But for many women, it really isn't easy. It gets a lot harder. And it's difficult to get really accurate data on that in relation to natural conception. So where we can get accurate data is from women having IVF, because we know exactly how many have those cycles, and of course their age when they have it done. And these are recent data from the HFEA that really just show how the chance of having a live birth from having an embryo put back. So that's actually at quite an advanced stage in the cycle, when you know that essentially conception has happened, goes from around 30% if you're under 30, right through to 15, 10%, down to 3 and 4% once you get into your 40s. And that's the chance of having a live baby. And of course, part of that, the loss there that is hidden by that graph is, of course, the increasing proportion of pregnancies that don't go through to a live birth, that end in miscarriage. And that, as you'll be familiar, goes up dramatically with age, from around 12% or so through your mid-20s, going up to about 40% at age 40. So a nice easy number to remember. And then going up to these really high levels once women get into their mid to late 40s. So there's really, and the biological basis for that is that so many eggs have the wrong number of chromosomes as you get older, and the stability of these chromosomes starts deteriorating, and that's what really underpins that. So essentially there is this real double whammy that you're facing with both reducing quality and quantity of eggs as you go through into your later 30s, when, as we saw, so many women nowadays are actually wanting to have their children. So what can we do about this? Well, potentially we do have a couple of options that are available here and now. And one that I'm going to talk a little bit more about is potentially freezing your eggs, as in this slightly glamorous advertisement, what can be described as stopping your biological clock. Potentially, you could actually freeze some of the ovary that contains those immature eggs. And Evelyn Telfer is going to speak a little bit more about that in her talk, I think. Or you could use someone else's eggs. You could use a younger woman's eggs. And again, this is data from the HFEA UK data. This graph here on the left is what you've already seen. But this is women who've used another woman's eggs to achieve a pregnancy. And you can see how the chances of success with that does not vary according to the woman's age. So this is all about the age of the egg, not the age of the womb that embryo is going into. So you can refine that, of course, because you can be your own donor, and that's what freezing your own eggs is all about. And this is becoming much more common. We don't have terribly up-to-date data in the UK at the moment. The most recent HFEA data only goes up to 2016. But what it shows is this increasingly steep curve of the number of women in the UK who are freezing their eggs. And then in the blue line, and then you can see in the two green lines, this quite big gap between the number who are actually coming back to use them. What we don't know from this data is why women are doing this. So this is for a mix of reasons. Some is for age-related fertility loss. Some is because they have a new diagnosis such as cancer where their treatment is going to compromise their fertility. And some are egg donors donating it altruistically for another woman. So we don't have that breakdown. But this idea of freezing your own eggs for later use has really attracting a lot of attention in medical and social circles at the moment. And just recently, there have been a couple of papers out on this, one from the Royal College of Obstetricians and Gynecologists that I was a co-author on, published earlier this year. And you may have seen in the news just a couple of weeks ago, this note from the Nuffield Council for Bioethics on this very same topic. Egg freezing in the UK is the title of it. So before we come on to some of the pros and cons, let's just look at the data of how good is this at actually making babies. And of course, we do have that big gap between the number of women who are freezing their eggs and the number of women coming back to use them. And this graph here is from one of the biggest published studies from Spain that report that from within one of the really world-leading clinics in this field. And they had 5,000 and a bit women who froze eggs, and 641 of them came back. And what you can see in the blue line in this graph is that if you're under 35 and you store enough eggs, you can get a really high chance of getting a baby as a result of that. So this is the cumulative live birth rate, and you can see it goes up to very nearly 95% chance of having a healthy live baby if you use enough eggs. And that magic number to get there is 24 eggs. So that will actually, of course, therefore, take more than one stimulation cycle of 2 weeks of injections and an egg recovery procedure to get there. The situation isn't so rosy if you're a little bit older. In the green, we have women over 35, and you can see that that doesn't go up so high, it isn't so steep, and that seems to plateau at about a 50% chance. And of course, older women will not only have less good quality eggs to achieve those pregnancies, they will be getting fewer eggs per cycle as well, that each stimulation cycle will result in fewer eggs for those women. So it's more difficult to accumulate those numbers of eggs. So why is this happening? Why are women wanting to do this? Well, the standard stereotypes that are usually trotted out here are the slightly cliched ones of women want to pursue their career. It's all about having fun before settling down. But actually, the research says it's not that. It's about their partner's choice is a very major reason for this. It is the wish for financial stability. And of course, in the moment, women are disproportionately taking the financial hit of the job insecurity that we're all going through at the moment. And the housing situation is also often cited. So we need to be ethical and rigorous about this. We don't really need these slightly over-glamorous, slightly trite adverts that you can see. And of course, these are actually American advertisements, they're not UK ones, but of course, that's perhaps just a matter of time before that sort of advertisement crosses the channel. So to counter that, one of the pieces of work that I'm personally involved in is trying to help women make accurate decisions about this, making the right decision for them. And that's a development of what's called a decision aid, which is rather fancier than just an information sheet, because it's really about helping a woman make her own mind up based on her values, her beliefs as to what she really thinks is the right thing to do for her, rather than just describing the options. And Michelle Peet there, whose email is there, is the lead investigator in this in Melbourne. So what conclusions can we come from these two reports about this? Well, they paint a fairly positive view overall, that this is something that can be a positive thing for women, but they both came to very similar conclusions that this is essential that women doing so have a very clear idea of the likelihood of success, the costs, and potentially the risks as well. Those success rates are going to be limited in women who are already in their late 30s, which is where the age at which women are currently doing this. And part of the problem here is the legal limits on duration of storage of eggs are challenging. You can only store eggs for this reason for 10 years at the moment. That's the law, and that's something that has no biological or medical basis and really needs to change. The Nuffield group also discussed the issue of egg freezing as an employment benefit that you may have come across, and that certainly is not an all benefit thing. That is certainly a double-edged sword, and really highlight the need for more research on that and women's actual experiences of using that as a potential employment benefit. But I think it can be a benefit, and under the right circumstances for the right woman, this can be something that's worth thinking about. So I hope that's within my 8 minutes, Sarah. Sorry, final thing I ought to just say, actually, we need to go back to the men, and I'm delighted that we got Chris talking about that, because actually, as I highlighted, one of the reasons for this is that it's often the men who are the source of the poor education, the poor understanding of this harsh biology that underpins this major problem.
Evelyn is Professor of Reproductive Biology at the University of Edinburgh and leader of the first team of researchers to achieve the full development of human eggs in the laboratory, and it was an amazing breakthrough. So over to you now, Evelyn.
Thank you very much, Sarah. So Richard has introduced the area very well, and I'm going to talk to you about some of the new experiments, new techniques that we're trying to develop. I do have a slight problem in that my slide won't change, Sarah. Apologies for this. It worked okay. I'll try and go here. Okay, so Richard has introduced the concept that human eggs are formed before birth, and you have this non-proliferating pool of immature eggs that must last the woman throughout her reproductive life. And these eggs are used throughout life. So they have these non-growing eggs within primordial follicles that are then activated to grow and will eventually form mature eggs that will be ovulated. Now, Richard also mentioned that because these primordial follicles are really quite vulnerable and they could be subjected to damage, particularly if a woman is, say, getting chemotherapy treatment, there's a technique now where women can have pieces of their ovary removed before they undergo certain treatments, and those pieces of ovary can be frozen, and these biopsies, the ovarian cortex, contain predominantly immature eggs. So these primordial follicles are contained within these pieces that are frozen for subsequent use. Now, of course, transplantation is really not the option for every woman, but it would be a valuable addition in terms of extending and preserving fertility to be able to grow these immature eggs entirely outside the body and obtain mature eggs that we could then fertilize. So that's a challenge that we've been trying to achieve results with growing eggs outside the body. So what I'm going to briefly talk about is two parts of this process. So lab-grown eggs, or in vitro gametogenesis, or in vitro growth. And there's two parts of that are developing the eggs that we already have, these immature eggs, growing them to maturity entirely in the lab, so growing our old eggs, or forming new eggs from stem cells, so making new eggs. So the lab-grown eggs that we've been working on for some time, we can take small pieces of human ovarian cortex that's obtained at the time of fertility preservation, but also women donate this tissue at the time of caesarean section, and we also have gender reassignment patients as well. And we can activate these immature eggs within the lab, and we can then get many growing eggs. So you can see here, this is tissue that has been activated, and we can then isolate these growing eggs to grow them further to a point where we could potentially mature them, and maturation meaning that they resume meiosis and they're at the stage that we would normally see at around the time of ovulation. So what we're able to achieve is these more developed follicles and eggs, which we can then grow for a further period and then try to get these eggs to reach the mature stage, similar to what we would see at ovulation. And we can get a proportion of these eggs to that point, and they reach the stage that we refer to as metaphase 2. So that's been a big achievement. And we now have this multi-step culture system where potentially we have eggs that we could fertilize, but we need a license to do this, and that's what we will be applying for from the HFEA. But we are improving our system to give us a better chance of getting good quality eggs to test their fertilization capacity. So we've used this multi-step culture system to make many studies, and we've got lots of publications looking at the effect of age, the effect of chemotherapy, and the role of signaling pathways in the development of these eggs in vitro. But we're now using this model system to study the potential to make new eggs outside the body. So as you heard from Richard then, females are born with a fixed number of these immature eggs, and that number declines as the woman ages. And just before menopause, there's a really low number of these immature eggs present. Not none, but some, a low number. But the question that we've been asking is whether these eggs or these ovaries have the capacity to make new eggs. So are there germline stem cells, so stem cells within the adult ovary that have the potential to make new eggs under the right conditions? And this question was answered in 2012 in a Nature Medicine publication by a group in the United States that showed that a cell type could be isolated from the adult human ovary. And these cells, under the right conditions, could form new eggs. So what we've been doing now is isolating these cells. So these ovarian stem cells or oligonial stem cells, and isolating them and mixing them with other cell types, somatic cell types. And you'll hear more about that from Chris about germ cells and somatic cell mixing. And we've been forming these mini ovaries. So this is our aggregates that we form, and we form mini ovaries entirely outside the body. And within those mini ovaries, we get the formation of follicles with eggs in them. So that's in a relatively short period of time that we're able to achieve that. And when we look at these eggs have markers that are similar to the eggs that you would see developing in the natural process. They have a marker called GDF9, which is a marker of oocyte or egg development. So that's indicating that indeed when we form these mini ovaries, we can indeed produce new eggs outside the body. So in summary, we can take immature eggs that are within the human ovary and we can grow them to maturity outside the body, so in vitro grown. We can also make new eggs or egg-like structures from stem cells when we combine them with the right somatic cells. Of course, we need to robustly test these human eggs, the ones that are grown in vitro or developed from stem cells, to determine whether they're normal and capable of being fertilized and forming embryos. So we still have a long way to go before any of these techniques can reach a clinical application. But what we have to remember is that these lab-grown eggs have many applications, not just in terms of direct clinical application, but in terms of basic science in allowing us to understand more about the human egg development, fertility preservation in cancer patients and other patients. And of course, these techniques could be the next generation of in vitro fertilization. But they're also applicable to animal production and in the conservation of rare breeds. So there's a long way to go, but these techniques have lots of potential. And finally, I would just like to acknowledge the people that do the work, the patients that give us tissue, the clinical collaborators, and the organizations that give us funding to carry out this type of work. And thank you for your attention.
So our next speaker is Chris Barratt, and he is the Professor of Reproductive Medicine and Head of Systems Medicine at the University of Dundee. And he's a clinical scientist at NHS Tayside.
I think it's important. This is going to focus on men. So we've had two discussions about the female. This is purely on men. So just to remind us that actually men are composed, as in females, are composed of somatic and germ cell tissue. And this is a picture to try and highlight this description. This is a picture of Marlon Brando, who was a film star many years ago. This is him when he was a wee kid, I think in Waterfront or something like that as a film. And this is him when he's a wee bit older. And what you can see in this translation, this is the same person, is that actually there's been a significant difference in the somatic tissue with aging, considerable difference. Of course, and the question then would be, is the reproductive capacity of the man changed with age as it has with the somatic tissue? And of course, that's not always easy to discriminate, because it's not always that you seem to grow older as you change chronologically. As you change your actually physical appearance. And an example here is, of course, Tom Cruise, who in 1992 and 2001 appears to look the same to me. I can't notice a significant difference. Unfortunately, my wife reminds me that actually I'm the same age as Tom Cruise and I don't look like him. So that's a bit of a disturbing event. But the somatic tissue here doesn't appear to have changed with age. The question is, has the reproductive tissue changed with age? And of course, there are other extreme examples of somatic changes in men. This one, for example, comes from Keith Richard. I couldn't get a picture of him when he was old or young. This is the only picture I can get of Keith Richard at this stage. So, and this is a question of, there's a development of age as a somatic tissue, and do we have an equivalent development as we've seen for females with regard to germ cell tissue? So the 4 points that I wanted to try and address today was, is there actually a problem here? For example, is the age of the fathers actually increasing? Is this an issue? And we've heard that there's an increase, for example, in female age at first birth. Does the reproductive capacity of men change with aging, or does it remain exactly the same? And if the reproductive capacity changes, what are those consequences of that change? And if they do change, what can we do about it, for example? So if we deal for the first question, in almost every database you look at, and this is from Australia, what we see is that there's an increase in the median age of fathers as time progresses. So in Australia, it's just under 34 in the year 2016. And whereas in 1976, it was about 28 and a half. So there's a substantial increase in the age at birth for the men. And that is a universal event. It's the same in the UK, it's the same in America, etc., etc. So there clearly is an issue of men being older when they're achieving conception. But the concept is that actually men can achieve conception very old, for example. So the question is there any effect of paternal age on reproductive capacity? And actually, there is a significant effect on reproductive capacity. And these are just some of the examples that I wanted to show you. So this paper is from Chris Ford. It was done some time ago. But what this basically shows is that if you look at men, and this is natural conception, if you look at couples where the man is under 25, and you compare that then to a man of age 35 to 39, then you see a significant difference, almost a 50% difference in the chance of conception over time in those couples. So male age has a significant effect for conception in vivo. If we look at just standard semen analysis and sperm function testing, there are a plethora of studies which show as a man ages, there is a decreased number of sperm, a decreased motility, an increased number of abnormalities, and as Denny Sackers has shown very recently, a substantial increase in DNA damage of the sperm as men grow older. If we look at assisted conception data, they generally show in large databases that there is an effect of age over time for men, independent of other variables, although the effects are relatively modest when it's taken into assisted conception. We also have data on a potential higher risk of preterm birth and low birth weight and increased morbidity in couples and this was on a 40 million database from Michael Eisenberg from Stanford, as a higher risk of premature birth, for example, as the male age, paternal age increases. And there are a plethora of other studies showing an effect on reproductive age of the male, from sexual activity to libido to potential endocrine changes in the man. Although, can you then change that and treat that using, for example, testosterone replacement is an age-old question. So we've noticed that so far that the men, that increase age with time to achieve a conception. We've noticed a negative effect on their reproductive capacity. But what effect does this actually have? Is there any consequences to an older man conceiving children? And probably the two most important aspects of that is being the so-called relationship between advanced paternal age and disorders such as autism and schizophrenia. And this relationship has been discussed and known for some time, and there's been a lot of data come quite recently to push forward these ideas. So for example, this was a very recent paper published which talked about that a 5-year increase in paternal age was associated with a 9% increase in odds for autism spectrum disorders in children. But even more remarkable, at least when I was thinking about how paternal age affects conception and the children, is the whole concept of the older fathers achieving conception with the children, and the children actually have a reduced longevity and a lower reproductive capacity. And this particular experiment has been done over 4 centuries, which is quite a long time in aging terms, and in 4 different populations. So this talks that the older the man, it gives a reduction in offspring survival and reproductive success of the next generation. So that is a substantial fitness effect, as they call it in biology terms, and the next generation as from the older man. So if these effects are there, and if we accept these ideas, what actually can we do? Well, I think a very important concept is we need to educate people. Many people understand as women grow older, there's a change in their reproductive capacity, but very few people understand that there's a change in the male reproductive capacity with time. And almost none of the students, for example, who we've talked to, where we ask them to draw a graph of time versus reproductive capacity in men, actually manage to make anything that's like a reasonable graph. And that is actually shown in recent data. It's published in Human Fertility, where 90%, greater than 90% of university students significantly underestimated the effect of male age on fertility. So one of the most important things we can do is to educate people about these effects. And then if we do that, that's a great starting point, because at the moment, the knowledge base of even relatively educated people, if you think university students are genuinely educated, is that they do not understand the negative effects of age on male reproduction. We absolutely have to get away from this idea, for example, where we have here, this is Mick Jagger, and he's achieved a conception later on in age. The concept then is that actually men, older men, and 72 I'm classing as older for this example, are actually achieving a conception, therefore everything's fine. And that's actually the message people are getting all the time. And what if you're looking at Guinness Book of Records, apparently the oldest man to achieve a conception was significantly old in Australia. But that is the exception to the rule. As men age, there is a significant negative effect on the reproductive capacity. And just using 1 or 2 examples of old men achieving a conception is not helping the image and our understanding of the biology and the messages to the public. So my last slide says basically very simply that actually we understand now, if we look at Marlon Brando, that the reproductive biology of your sperm cells of this man represent and look effectively like the somatic tissue. It is not this situation where it remains the same. The reproductive capacity of men significantly changes with time. So the message is, men, don't be fooled, your germ cell tissues do age, and they have significant consequences for the children.
We have our final speaker next, Dr. Vasanti Jadva. And Vasanti is Principal Research Associate at the University of Cambridge's Centre for Family Research. And that is where she researches the psychology of children, parents, donors and surrogates in relation to assisted conception and family arrangements.
So I'm going to be talking about the relationship between parental age and children's psychological development. So we've already seen similar tables in some of the other presentations, which is showing that the sort of live birth rates of parents in England and Wales has been going up. This graph here shows data from 2017, showing that around a third of live births was to parents aged between 30 to 34 years. So as we've been hearing, there have been a number of studies that have looked at the relationship between parental age and children's psychological development in the general population rather than specifically assisted reproduction. And those studies tend to show some of those studies tend to show poor outcomes for children of older parents, as we've just been hearing. Others tend to find no differences, and some tend to report beneficial outcomes, particularly in relation to older mothers. So studies tend to define older parenthood differently, which does make it really difficult to compare across studies. And also some of the larger cohort studies can't always identify the reasons why you find these differences. And relatively fewer studies have actually looked at the impact of older parenting on older children. So a lot of the studies have looked at children who are infants or sort of up to middle childhood. And women having children older may also experience fertility problems. They may have experienced a greater weight in order to become parents. They may have undergone psychological strain or fertility treatment and also needed to use donor gametes. So what impact does that then have on the psychological well-being of the children? So one of the only studies that has directly looked at the relationship between parental age and child adjustment has actually been conducted by Jadva and colleagues and published in 2009. And in that study, they had a sample of 642 mothers, 439 fathers, and they divided the group up based on the age at which the mother became a parent. So below the age of 31 years, between 31 and 38 years, or over the age of 38 years. And the children were 4 to 11 years at the time that they collected their data. And what they found was that mothers and fathers from the older age group reported more depressive symptoms compared to the younger parents, and also that there was less expressed warmth between the couples in the older age group compared to younger parents. But in terms of the children, they found no differences between the groups on conduct problems, peer problems, pro-social behaviour, depressive mood, anxiety, and physical symptoms. The other way in which we can understand the relationship between older parents and children's psychological adjustment is by extrapolating from the studies that have been conducted on assisted reproductive families more generally. So this is one of our studies, a longitudinal study of families where a child has been conceived through surrogacy, egg donation, donor insemination, and also natural conception group. And in this study, we have parents who became parents at an older age, particularly when they had used surrogacy or egg donation compared to the other types, donor insemination or unassisted conception. And when we last saw them, when the children were age 14, we found no differences between the groups in terms of their child adjustment, child well-being, or child self-esteem. One of our newer longitudinal studies is looking at the outcomes for families who have used IVF using an identifiable egg donor. And in this study, we have 85 families who had used IVF using donor eggs, 65 families who have used IVF using their own gametes. And families have been seen so far when the child was aged 1 and more recently at the age of 5 years. Now in this sample, we have parents who became parents over a broad age range. So for example, for the mothers, the youngest parents were aged 28, the oldest 52. For fathers, they ranged in age from 29 to 62 years. So what that means is we're able to look at this data by splitting it in terms of parental age. So if we do that and we split the data looking at these sort of 4 age categories, so those below the age of 35, those aged between 36 and 40, between 41 and 45, and over the age of 46, this table here shows us the relationship between mother's age and whether or not the couples had used donor gametes. And as we can see, the use of donor gametes increased. So all of the mothers who took part, also all of the families in our study who took part, where the mother had a child over the age of 46, had used donor eggs. And that study has found that at the age of 1, we found no differences. The only difference we found was that older mothers reported lower perceived social support. At the age of 5, we found that older mothers, so those mothers who had a child after the age of 46, reported poorer relationships with their partners compared to younger mothers, so those who had a child under the age of 35. Older mothers and also older fathers reported greater parenting stress than younger parents. And also we found the correlation between these two variables. So those parents who reported poorer outcomes in terms of their relationship quality also tended to report greater parenting stress. When it came to the children, however, we found no differences in children's adjustment using questionnaires completed by parents, and also no differences in child anxiety and depression using a standardized puppet interview that we carried out directly with the children themselves. So one of the things that we've been doing in this study is interviewing parents about their experiences of parenting, as well as their experiences of fertility treatment. We didn't ask parents specifically about their experiences of being older parents, but it was sometimes something that came up during these interviews. So, for example, this mother said, in relation to telling her child about the use of donor eggs, I've started to think about that in terms of telling the child, I do want him to know. I do want him to know. I don't want it to be avoided. And again, from the point of view that being an older mother, you know, should anything happen to me, I wouldn't want him then to find out afterwards and feel angry and annoyed about that. Other parents mentioned their age in terms of having a sibling for their child, or wishing that they'd had their children younger. So for example, this parent said, I think the fact that we're older, tireder, I wish really, you know, I wish I'd had them 10 years ago. And another said, I just wish we'd been given the opportunity earlier in terms of testing that they'd undergone during fertility treatment, because there could have been more than one, and there is. We've got a frozen embryo, but because of our ages, and I was quite poorly after I'd had the child, not mentally, physically, because I'd lost quite a lot of blood, we made the decision not to put it back. And other parents mentioned sort of the advantages, I guess, of being older parents, including having more time, for example, to spend with their children. So for example, this parent said, I feel that I'm doing an awful lot of things other parents don't do. Either they just don't have the time because they're working all the time, or so, you know, we do have nice times and fun together and stuff. But yeah, I guess, you know, I'm not a sort of 20-year-old who's going to take her to a disco when she's 16. So overall then, from the studies that have been conducted, although not very many studies have looked specifically at the relationship between parental age and child outcomes, studies that have looked more generally at assisted reproduction families, which tend to have parents who are much older, have found that actually there don't seem to be any problems in terms of children's adjustment, at least in terms of the studies that have been conducted so far. However, we do need to have more studies looking at much older parents. So many of the studies that have been conducted so far tended to have parents sort of having children in the late 30s or mid 40s. But what is the consequences when they're actually having children much older? So sort of in their 50s, for example. And we don't know as much about those sorts of families. We also need more research on the impact for older children. So like I said earlier, a lot of what we know is about the outcomes for children up until the age of sort of middle childhood. But what happens when they get older and are therefore being parented by older parents? And also most studies have reported data from the parents' perspectives on child adjustment. So we really need more studies using data collected from the children themselves in order to get a better understanding of what having an older parent is like for them. Thank you.
I can see that we've got lots of questions in the Q&A. So Richard, Evelyn and Chris, I hope you're joining us again, and we can start on some of those questions. And I'm going to endeavour to direct the question to one of you first, and then allow the rest of you to chip in. So audience, please don't forget to keep posting your questions in the Q&A, and to say if it's for a particular speaker, and also to vote for the questions that are already there. So I don't think anyone's been voting terribly much this evening. So I'm going to go from the top of the questions and see what questions we had for Richard. So I think this is something that I was thinking about asking as well. And it's from Steve Galatley. Apologies if I've mispronounced your name. So Richard, it seems that this isn't just a problem for scientists and clinicians to solve, but also politicians and other social organisations. Are there any steps taking place to try and raise awareness of underlying biology to policymakers, and to try and stop healthy women being in this position in the first place? So if you want to have a go at that, and then I'll come into the other panelists.
Yeah, that's a great question, and actually, it would be a great topic for a whole pets session one day. Of you excuse me, being a clinician, I take a medical approach to it primarily. I'm not really qualified to take some of the other things, but you're quite right. This is putting a sticking plaster on a bigger wound. And I absolutely agree that this is a prime example of where prevention is better than the patch-up cures that can currently be offered to it. I think what we can do is we can produce documents such as what I was involved in from the Royal College and the Nuffield Council for Bioethics, similarly, which are important documents that one hopes that government or the Department of Health at least takes some notice of, and it raises public awareness that these are big societal issues. The one perhaps concrete thing I think where things have changed actually is in the provision of education. And again, the RCOG in combination with the British Fertility Society has actually a fertility awareness campaign that's been going on for a while now, and has actually produced videos for that, for particularly aimed at adolescents, to educate younger boys and girls or young adults about reproductive aging. And I think educating the males as well as the females is absolutely critical for that. So that is actually now becoming incorporated into school curricula. So I think there really is actual progress going on there through that initiative from the BFS and the RCOG. But yeah, absolutely, this is a big societal issue rather than just medicine picking up the pieces.
Chris, would you like to chip in on this one? I'm sure you have some views.
Sorry, well, I think the idea of society not understanding their reproductive capacity is just, I think, with regard to males, it's blatantly obvious. I mean, it is a massive problem with men understanding their reproductive capacity. They just see pictures of Rod Stewart, Mick Jagger, or Charlie Chaplin, and having a conception, and then everything's okay. And I think that also places an unfair burden then on the woman as well. So any education that we can achieve from young teenagers onwards has got to be a very positive thing. And quite frankly, we just don't do enough of it. So getting an RCOG, BFS type collaboration doing this work is just fantastic.
Great. And Vasanti, how about you? Is this something that you want to comment on?
I would just agree that, I don't think people really do understand about the kind of fertility decline. I think there's a number of people that have been bringing that up for some time now. I guess in terms of the studies that we've been doing, we tend to see people who are having difficulties in actually having a child and needing fertility treatment. But also in terms of our single parents who have used donor sperm, for example, I'm sure, had they been able to have their children earlier because they were in those situations, and then they, had the support and everything, then that would make a difference. We know, for example, that the relationship between financial stability does have an important impact in terms of children's psychological outcomes. So those sorts of factors would make a difference.
And Evelyn.
I think there's also an issue that people feel very young when they're 40 these days compared to what they used to. And that doesn't help, to be honest, does it?
Yeah.
Evelyn, is there something that you wanted to add?
Yeah, sure. I mean, one of the things that I've noticed when I've been doing public awareness lectures, that young women are really quite surprised when they hear about this, kind of rapid decline in their fertility. But there's a lot of education programs going on, and I would mention Joyce Harper in London, who's been doing some excellent work in terms of outreach. So I think it is something that we need to get the message across. But I think there's also, in terms of like the basic work that we do, many people are very interested because they see that as the panacea. That's that is going to help everyone. And as I said, all of these techniques are going to take a long time to really be sure about.
Yes, and I think there's sort of as well this sort of reliance on medical interventions.
Yeah.
And of course, as we all know, it doesn't work for everybody,
Yeah,
and
absolutely.
is absolutely heartbreaking when they have repeated failure. So the next question I'm going to take is actually for you, Evelyn. And this is someone who's chosen to remain anonymous. So out of interest, are we bypassing selection processes of egg quality by maturing a number of eggs?
Yeah.
And is there any evidence out there in your lab or others of similar fertilization capabilities or other markers of quality, aneuploidy, etc.?
Yeah, sure. I mean, that's a really excellent question, because obviously within the ovary, there's only a very small number that would ever be selected for ovulation. And what we are doing is indeed bypassing any checks that may be there. Now, we do know that in the young ovary, there's a lot of eggs that are actually just, shed from the ovary before you even reach puberty. But what we don't know if they're bad eggs or if they do have the potential to grow. So we have to look at animal models. And the only model that we have at the moment where we can grow eggs outside the body all the way from the immature stage, get them fertilized and produce live young is the mouse. And we can produce live offspring from the mouse model from the most immature stages. Initially, with the early work indicated that there may be some problems with the offspring, but then there were changes in the development of the techniques, and now that does not seem to be such a problem. So I think there's a combination of factors about the intrinsic developmental potential of the egg, and also the factors that we subject them to. And we have to get the right balance, and we have to then, work out good markers of quality. And we're now embarking on animal work, actually a sheep model, and we're now growing sheep immature eggs to a point where we can fertilize them and then transfer them. And we will be studying these embryos and the live offspring, if we get them, in great detail. So we might be in a better position to answer that question. But it is an important question, and it's one that we don't fully know the answer to.
Thank you very much for that really detailed answer, Evelyn. I'm going to turn now to some questions for Chris, and we have two. I'm hoping that I can ask them both at the same time. So Annabelle Slater has asked about the study about children born to older fathers having lower fitness, and she'd like a bit more detail about that, please, the sort of controversies about it and any possible explanations. And then the other question, which is from Georgios Krontiris, and that is, do you believe it's important to tell young men the value of fertility preservation? So I suppose this would be like, we've been talking about egg freezing for women. Should men be sperm freezing? And is there enough data available that would justify that as a
okay, I'll probably try and answer the last question first, really. Obviously sperm freezing has been around since the 1950s in humans and very successful. So the idea could be that you may get men who want to preserve their fertility to store sperm, say between the ages of 18 and 25 as an example. So that's a possibility. That seems to me rather extreme to do that. It's quite costly to do that. That has been proposed on a number of occasions, but I don't really think that's quite the answer to do that. Although it is technically possible to do that, and it's possible for men to do that, of course. But I don't think on a global scale that's a very intelligent way to go about it. I think the real question is, as we've said, driving home the messages to couples and to men. And I think what we need to understand is why don't couples get these messages at the moment, despite a plethora of just vehicles to do that. So I think that's the answer to the second question. The answer to the first one is much more complicated in reality. These studies of looking at the fitness of the next generation have been done in many animal models, as an example, from worms to higher animals. and they've shown that the paternal age has an effect on the next generation. And the idea behind that, to some degree, is the damage to the male germ cells as the man ages. And the concept is that there's a higher level of damage to the germ cells in men who are older compared to men who are younger. And the concept then is that that damage is then transmitted to the next generation, which may have consequential effects to the viability of those offspring and the reproductive capacity of those offspring. So that's one of the concepts that people are looking at, and there's significant evidence for that, although there are some counter ideas and hypotheses related to that. That particular study that's looked over 4 centuries, of course, was an observational study, looking at data that was already there. I don't think many people are 400 years old. But basically, that study was done from records, etc. And what it was trying to do to some degree was to see in humans where we're seeing the same observations as there are to be, and to some degree, as they were seen in animals. And that's probably the biggest study using that type of data. And because it used different data sets from different generations and centuries, then the concept is that that data is relatively strong, although the paternal effect is relatively weak. It's significant and it's there, but it is not, it's not like a man of 50 who produces children and they basically all the children die young. It's not that absolute effect, but it is a significant, and it is an observable, repeatable effect from that data.
Thanks, Chris. The next question that I'm going to take is from Rabina Butt. For a single 42-year-old who's never conceived naturally, is freezing eggs, not embryos, still a viable option in the UK? And what's the likelihood of a successful embryo being created and a live birth for a 42 to 45-year-old? So I think perhaps that's one for you in the first instance, Richard.
Yeah, thanks. It's viable insofar as it's legal and possible. You will find clinics that will be happy to do that for you. But I think it's fair to say, and I'm sure you would be told if anyone who went ahead under those circumstances, that the chances of success of having a baby from doing so are going to be very modest. You saw in the graphs I showed that the success rates of an IVF cycle, creating an embryo and then putting it back with fresh in the UK in women who are aged 40 to 45 is only about 3 to 4% per embryo that's put back. Of course, if you try and do this, then you're likely to get a number of eggs and therefore perhaps a number of embryos, but that number is going to be small if you have one cycle of ovarian stimulation. And so to build up lots then gets very expensive indeed, essentially, because this is not something that, of course, the NHS is going to be providing. So it's technically possible. Whether it's viable depends on whether you think that a low single figure percentage success rate is appropriate for someone who wants to do this.
And I think this sort of highlights something that that certainly we've tackled at PET, and that I was pleased to see in the Nuffield Council of Bioethics briefing, is that there needs to be better information for women about these chances, because we sort of have a sort of poor understanding of chance, otherwise we wouldn't buy a lottery ticket from time to time. There's very little chance of winning, and the odds of these sometimes for some of these women, particularly the older women who are working, are incredibly poor, and I don't think always well explained from what I've heard from some women who have frozen their eggs in their 40s.
And I think the other aspect of that is that the data you see published in scientific literature is from the best labs. So that graph I showed you from Ana Cobo in Valencia, her lab is absolutely one of the best in the world. And your local clinic may not be up to that standard. So you need to not just say what's the best in the world, but what actually can you do to offer me this treatment?
Yes. So I'm going to move on now to the next question, unless Evelyn, because I could see you nodding, whether you were just agreeing or had something else to add.
No, I was agreeing.
Great. Another question from Annabelle Slater, and this one's for you, Evelyn. This discovery that new eggs can be made must have rocked the scientific community. What do we know about how different these eggs are and how differently embryos may develop from them?
Yeah, absolutely. So it did indeed rock the scientific community, and it continues to rock the scientific community. And when this observation was first made in mouse, it was almost unbelievable. And I was one of the people who just could not believe it. And I really became convinced through meeting with the scientists who had carried out the work in the mouse, and then seeing for myself some of the experiments. So I moved from very much from a camp who thought, no, this can't be possible, to, yeah, let's investigate it further. And it continues to be an incredibly controversial area. So the idea that you can get cells within the adult ovary, and particularly the adult human ovary, to make new eggs is, something that no one really thought possible. But I think you have to separate it from cells being present, because we now know that adult stem cells are present in almost every single tissue system. But what these cells probably do not do, they don't contribute to the normal physiology of the ovary. So they probably are not making new eggs under normal conditions. So what we're looking at is a way of tapping into these cells to make new eggs. Now, what are these eggs like? Well, the real answer is we don't have enough information about them yet. We have information on mouse where they have the potential to form, embryos and live young, but we don't have the volume of studies or the longitudinal studies to answer that particular question. And we're at a very early stage in the human work. So I think it's clearly something that we're hoping to look at in more detail, and hopefully more groups will look at it as well.
Great, thanks for that. I was sort of looking because I'd seen a question for Vasanti, and I've managed to lose it in this infinite scrolling of all the questions that I've now got before me. So while I'm looking for that, there's a question for all from Rachel Bowlby. It's surely difficult to evaluate or otherwise experiences of older parenting, given the ongoing shifts in the norms of parental age. In other words, to be a new mother of 40 in 1980, or let alone in 1960, was something quite different from 2020, when it's not that unusual. And similarly, young parenting under 20, say, is now semi-demonized, but was normal quite recently. So I don't know whether you want to start on that one, Vasanti. It wasn't a question I had in mind for you, but I think it's a good one for you to get your teeth into.
Yeah, I mean, that's kind of what I was mentioning in my slides as well, is that the kind of how studies define older parenthood differs from one study to the next as well. So, and part of that reason is because of societal perceptions of older parenting has changed. So, I mean, the fact that the 2017 data from the ONS shows that live births are to parents aged between 30 and 34. That's very different to what it was, going back 10, 20 years. So it's a good point. And I think that's one of the reasons why perhaps, families who have a child in their 40s don't perhaps experience stigma and all the negative consequences associated with stigmatization, because it is a lot more normal now to see parents having children at that age than would have been so in the past. So yeah, it's a good point, and it does make things difficult when you're trying to look at the literature on this.
Yeah.
Can I comment?
Yes, I thought you might want to come in, Richard.
It's quite interesting, actually. If you look at those graphs that I showed from the ONS that Vasanti has just commented, if you actually take the timescale back a bit earlier to sort of from the 40s through to now, you actually get rather a different picture of actually some of these rates of childbirth in women now are actually much more similar to what they were in perhaps the 50s. But I guess, and then it showed a big drop with the advent of modern contraception, and then it's picking back up. But I think one of the big differences, of course, is that now that might be a woman's first child at that age, whereas historically that might have been some children down the road, because that's what those graphs are not of first children, they're just a proportion of women having a baby at that age.
And I suppose also there was a huge disruptive factor of World War II that would have had impact then as well, with the men being away and men not coming back and women perhaps remarrying, etc. And I think that it's sort of interesting when you sort of think about this, and I know I'm sort of making it about myself again, sorry, but thinking about my own mother, who always told me that her age was 21, and until I had any rudimentary grasp of maths, I was totally confused by this, because I had a brother who was 28, and it was only when someone told me this is biologically impossible that I started to ask questions at home. So now I've got another question here, and this one is from Mark Hamilton, and he wants to know about screening for individuals at risk of premature ovarian or testicular aging, and he wants to know, is this a dream or will it be reality? So I don't know who wants to go on that first. Do you want to try that one perhaps first, Evelyn?
Screening for aging?
Yes, for premature ovarian aging, and perhaps we'll let Chris deal with testicular aging.
Yeah, sure. I mean, clearly, that's one of the things in terms of being able to develop, understanding human oocyte development, that our culture systems might allow us to find out greater markers of aging, because we're able to look at a range of material from different ages and different physiological conditions. So at the moment, of course, there's there's things in terms of meiotic markers and so on, but I think that's something that will be more fully developed when we have better systems to study human egg development. But I think Richard's probably more qualified to talk about the clinical markers that are in place.
Richard, do you want to chip in then while we're talking about the female before we go to Chris?
Okay. I think it's very much a hot topic, actually. It's an area of active investigation to try and identify markers of reproductive aging in women. Clearly, someone who is destined to go through an early menopause might want to know about that in advance. And when sometimes you can see damage that is going to occur, and perhaps most obviously in a cancer patient, then clearly you can do something about it. But the reality is, at the moment, it isn't possible to do with accuracy. And actually, in many ways, the best way of knowing the age that you're likely to go through the menopause is to ask your mum what age she did it. And that gives you a pretty good clue as to the likelihood. Of course, if your mother went through the menopause at 30, then you've got a pretty good pointer that you may be at risk. And under those circumstances, perhaps you can have some tests that can identify things, but they can only identify damage when it's already essentially well established, rather than identifying it before it happens, which of course would be the holy grail. Yes.
And Chris, do you want to come in on the male element here?
Yeah, I think that the advantage of the men, of course, is then you've got a semen sample you can look at. So if you've got azoospermia very early on when you'd expect to have sperm, then that is, of course, a marker for that. The question then, I guess, is can you are there any techniques to actually either store cells or actually grow cells in vitro to try and counteract that? So there have been studies in animals where you can get cells out of the testis, grow them in vitro, and then inseminate an egg and then achieve a live birth in animals. That's not been done in humans. But what we do know now in humans over the last couple of years is that actually you can really go down in spermatogenesis to the round spermatid stage, which is the first haploid event in spermatogenesis. And you can inject those round spermatids into eggs in humans and achieve live births. And up until a few years ago, we thought that was completely impossible. So you can, if you had a man who prematurely aged or there'd been an early aging event and there was a block in spermatogenesis at that level, in theory, you could get those cells and inject into eggs. So there has been some progress in that way. But in vitro generation of spermatogenesis in humans and then achieving healthy live births is still a bit of a long way off, I would suggest.
Thanks, Chris. I've now found the question for Vasanti, which I'd lost. Vasanti, what do we know about the outcomes for young people who due to having older parents, may need to care for them due to their parents' ill health when they're young, or perhaps having one or perhaps both parents dying when they're teenagers or young adults?
Yeah, so like I said, most of the studies that have been done in this area have looked at younger children. So therefore, in relative terms, they tend to have younger parents. So we simply don't have enough studies out there looking at the consequences for older children or even young adults of having older parents. And certainly that there is a chance that their parents may be unwell, for example, or they may lose their parents in older childhood, depending on the age of the parents, of course. So, but I mean, this, unfortunately, the answer is that we don't really know very much at all about that.
Okay, thank you for that. Now, gosh, I'm torn now because we've not got much time. I think I'm going to go for from another one well, actually, this one's from an anonymous attendee. And this is something that was come up in our pet office, actually. For ladies undergoing IUI, is it better to select male donors that are younger in age, e.g. would a man aged 23 be more likely to result in a successful procedure than one aged 38, for example? So there we go, Chris.
Okay. I always say to students in the class that actually there's always the common concept of the older man and the younger female. I think what women have to understand, if they're growing older and they want a better chance of conception, then they should actually, all else being equal, I hasten to add, then it would be preferential to achieve a younger man as compared to an older man. And I think Chris Ford's study showed that for an in vivo conception. I think when it comes to IUI, the effective paternal age is slightly reduced in those types of studies. So there may be a significant difference there, but it wouldn't be enough to be able to cause, I think, a great deal of concern, because IUI allows you to enrich the cells and do repeated cycles, etc., etc.
Yeah, I think when it came up in the pet office, it was because we were talking about the whole the vast amount of information you can get about various characteristics of a donor, looks, hobbies, all those sorts of things.
Yes,
and
yes.
the geneticist in the office piped up and said, I just want the younger the better, because there's less chance of chromosomal abnormalities.
Well, I mean, you can always rely on a geneticist to actually put you straight, cut to the chase on things such as that, of course.
Yeah, his ability to write poetry didn't cut much ice there. So yeah, and I'm just going to see, there's one sort of quite long but controversial question, perhaps for you, Richard. And it says, I'm surprised that Professor Anderson answers this question: what can women do about reproductive biology, by suggesting freezing their eggs is the best option? Does he agree that doctors should still explain that the optimal age is 20 to 35 because the body ages and pregnancy complications, etc., and finding a young partner? IVF is an option, sure, but second best. IVF is not positive for women when the underlying problem is our unfair sexist society, but it's definitely positive for business, I think, which means the fertility business. So sort of a nice controversial one there for perhaps for you to end on.
Well, I think it's absolutely correct. I mean, reproductive medicine is a sexist business. You alluded at the beginning, though, Sarah, that there is an underlying biology, but clearly clinics offer this because they're not a charity. They're not there. They are there often to generate profits for their owners and shareholders. So that's why they offer this. They don't do it at a loss. And indeed, throughout production, there are plenty of other examples of the essential sexist nature of what medicine offers here. The most classic example, of course, is men with a sperm. Treatment for that is for their female partner to have IVF. They don't do anything other than produce a sample on the day. So that's perhaps the clearest example. So yes, I agree. I guess my answer was what can medicine offer in those circumstances, not what is the right way for society to be organized to prevent this arising, which, as I say, we discussed to some extent up front. So absolutely important question.